In this video, we're going to be covering Biology 30, mitosis and cytokinesis. This is going to be a review, which assumes that in your class you've already covered this topic. So, just to recap, cells in the cell cycle, there are two phases that the cell goes through. It's either an interphase or it's undergoing mitosis and cytokinesis. So just to brush up on interphase, it's split up into three distinct phases. So if we have our cell here, we'll give it its chromosomes, its full set, one blue from mom, one red from mom, and same thing for dad. So we call this 2N, one chromosome set from each parent. And the cell spends most of its life in the phase we call G1, when really it's just doing its tasks, whatever it's meant to do. So example would be a stomach cell or a skin cell, going about its business and doing what that cell is built to do. In G1, the cell basically just gets a little bigger, certain proteins get made to prepare it for mitosis, but the most part is it just gets a little bigger. Okay, so we can still draw our little chromosomes, just hanging around. The next phase, in interphase, is called the S phase. S standing for synthesis. And what gets synthesized is the chromosomes. Each of these chromosomes then gets replicated. So we'll draw that with what you might already be familiar with. Sister chromosomes. So each of these chromosomes have now duplicated itself, and they're connected to each other by this little protein in the middle called a centromere. Not to be confused with a centrosome, which are these proteins out on the side, centrosome, whose jobs are going to be to pull these sister chromatids apart later on in the cycle. After S phase, we go to G2, which really is just the cell doing a few last-minute checks to make sure that it can go ahead with mitosis. So, just to recap, interphase is just the cell going about its daily life, preparing for mitosis at some point. It starts off with a full genetic complement, what we call 2N, one chromosome set from each parent. It then moves on to the S phase, where each of these chromosomes then gets duplicated, creating what we call pairs of sister chromatids. See how the vernacular changes from chromosome to chromatids. It's just the way that we describe what's happening. It then moves on to G2, where the cell gets a little bit bigger again and prepares for mitosis, which we'll now cover. A lot of people have some misconceptions about mitosis and how it relates to cytokinesis. I'll explain what each of them actually means. So people tend to teach or think that mitosis is going from one full 2N cell to two daughter cells, each of them with the same genetic components as the first. This is partly true, except mitosis, what it really is going from a 2N cell, replicating the DNA inside of itself, and moving each set to opposite ends of the cell. The actual splitting part for this cell to start splitting apart into two separate cells is what we call cytokinesis. These tend to happen in the same process, usually at the very end of mitosis, but if you have a teacher who's a bit of a stickler for vernacular, you know that mitosis isn't actually the splitting of the cells. Mitosis is basically everything else. Now to actually go through mitosis and review it, don't forget mitosis is split up into 4 phases, usually remembered by the mnemonic PMAT: prophase, metaphase, anaphase, and telophase, at which point cytokinesis then occurs. So first we're going to go through prophase. So we start off with our cell, just as from before. Each of them has been duplicated already in the S phase, so it's ready to go. What happens in prophase, don't forget that everything that's happened in interphase so far has happened inside the nucleus, not inside the entire cell yet. But what happens in prophase is that the nucleus starts to break down, allowing the chromosomes to float freely in the cytoplasm, or most of what makes up a cell. And our centrosomes out on the side have now begun to put out little, what's called microtubules or microspindles, to connect to each of these chromosomes and to each other at some point as well. Microtubules, very easy to remember. Micro means small, tubule means small tube, so small, small tube. And each of these small, small tubes just comes from each centrosome, connects to the centromere of every chromosome. Remember to keep those straight. And these are the main events that happen in prophase. After prophase, we now move on to metaphase. Now metaphase, very easy to remember what happens here. This is basically just a checkpoint in which the chromosomes are all lined up nice and neatly in the middle. Our centrosomes are still off to the side here with their microtubules, or microspindles some people call them, connecting to each centromere. Very easy to remember because the chromosomes are all lined up in the middle. M for middle, M for metaphase. After metaphase, we now move to anaphase. So, anaphase is where most of the action occurs. So, we have our cell, our centrosomes, and finally the spindles have started to pull apart at the centromeres until each chromosome, they're now called chromosomes again. Remember how before we used to call them chromatids? Once they get pulled apart, we now refer to them as chromosomes again. And they each get pulled to opposite ends of the cell. This beginning of the pulling cycle is what we call anaphase. Compare that to telophase, which is where the chromosomes are almost completely at opposite ends. Of the cell, and each cell is basically ready to split. As well, if you start to see this, what they call a cleavage furrow, start to form right in the middle here, you know that the cell has started to undergo cytokinesis, which is the actual splitting of the cell. Cytokinesis tends to occur in late telophase. That's why people tend to kind of put them together. So if we now finish cytokinesis, what we're left with is two fully formed cells, each with a complete genetic complement. That's exactly the same as the mother cell that we started at the very beginning with. That is Biology 30-1 review, mitosis and cytokinesis.